Hello everybody, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati, and in this module we were discussing about the cell biology, as well as we are discussing about the microscopic tools what is available to perform the different types of experiments. So now in today's lecture we are going to discuss about the different experiments what you can perform with the help of the microscopy, and I have tried to make a problem like situation so that you will be understand under what problem you can be able to utilize the microscopy. So, let us start the discussion about the different experiment what you can perform with the help of the different microscopy techniques. So, our research problem 1 is very simple, that scientists have discovered a new bioactive phytochemicals from the neem tree. So, you know the neem tree, right, and now he wants to test its effects onto the propagation of the malaria parasite. So, what he wants to ask is that he has actually isolated a new bioactive phytochemicals, and now he want to test whether it is inhibiting the propagation of the malaria parasite or not. So, you know that the malaria parasite cause a disease called malaria, and if you go with the little background, you know that the malaria parasite requires the two hosts, one is invertebrate host and the other is vertebrate host. So, in an invertebrate host you have the mosquito which actually bites to the other vertebrate host, for example the humans, and that is how it actually goes from one body to another body. And within the invertebrate host they have a complete life cycle through which the gametes are fusing with each other to produce the ovum, and that is how they are actually producing the merozoites, and then merozoites are being so that is how they are producing the sporozoites, and these sporozoites are actually being injected by the mosquito to the human being, and then these sporozoites actually goes into the liver, and then it completes its life cycle within the liver to generate the merozoites, and these merozoites then infects the RBCs to form the different stages, and all the stages what is present in the RBCs are called as the arthrostatic life cycles. So, within the arthrostatic life cycle you have the different stages, like as I said, you know, once the merozoites are coming out from the liver. So, from the liver you are getting the merozoites, and these merozoites are actually infecting the RBCs, and then it is causing it is forming the first stage which is called as the ring stage. So, this is a typical ring stage what you see after as soon as the merozoites enter into the RBCs. So, why it is called as ring stage? Because it is having a ring like appearances where the nucleus is on the one corner and then the cytosol is distributed, and then ring stage is getting converted into the trophozoite states, and then trophozoite is actually converting into the schizones, and the schizones are actually releasing the new merozoites, and these new merozoites are again infecting the new RBCs. So, if you see the life cycle, what you see is that the merozoites are infecting the RBCs, and these RBCs are then forming the ring stage, and after the 10 hours the ring stage is getting converted into trophozoite stage, and then from trophozoite it is actually causing the production of the schizones, and then from the schizont it is actually again releasing the. So, once the RBCs containing the schizones is going to burst, it is actually going to release these merozoites, and then these merozoites are actually going to infect the new series of RBCs. So, what we really want to know is that if we actually treat the parasite, or if we treat this particular culture with the phytochemicals, so whether it is actually going to complete its life cycle or not. So, experimental design is very simple. The experimental design is that you can actually take the ring stage parasites, and then you incubate that with the phytochemicals, and you ask whether it is inhibiting the propagation of or the transformation of the ring stage parasite to reach to the schizon stage parasites. So, typically what you have to do is you have to just first produce the ring infected RBCs with the help of the synchronizations, and then you incubate that with the help of the increasing concentration of the phytochemical or the test molecules, and then you incubate that for some time, and then after that you actually are going to make a smear after 34 hours to see whether the ring is being matured to give you the Chidon stage, because under the normal circumstances it is actually going to complete its life cycle, and that is how it is actually going to give you the Chidons within the 35 hours. If you want to study the reinvasions, then you again make a smear after 72 hours, and that actually is going to tell you whether your compound is also having an effect on the reinvasions. Reinvasion means the Chidons are going to release the merozoites, and these merozoites are whether infecting the new RBCs or not, because that is how it is actually going to give you the new rings. You can also ask whether the compound is the parastatic or the parasital, which means whether the compound is killing the parasite or whether it is simply stopping the growth of the parasites. That you can do simply by the drug removal. So, what you can do is you can treat the parasite for the drug molecules or the test molecule for some time, and then after that you actually can remove the parasite and keep it into the fresh media and let them to propagate. So, if the parasite is dead, it is not going to propagate into the fresh media, but if it is only stopping its growth, which means it is still be live, then it will start making the growth. So, that is what you have to do. When you just remove the drug, you wash the parasite culture and then you put it into a new media, and then if you prepare the smear after 72 hours, if you see the viable parasites, then actually it is going to say that the compound what you are testing is the parastatic in nature, means it is actually stopping the growth of the parasite, but it is not killing the parasite. So, these are the three questions one can ask with the help of this microscopy based assays, where you can say whether the compound is killing the parasite or not, whether the compound is inhibiting the reinvasion event or not, and whether the compound is parastatic or the parasital. So, to performing these experiments, you require the media like the RPMI 1640 cell culture media, which we require to culture the malaria parasite. Then you require the Albumax 2, which is actually a powder which actually required for propagation of the malaria parasite. You require the 0.22 micron filters. You require the filtration units to prepare the media, you require the autoclaves, you require the vacuum pumps, and then you required a upright microscope so that you can be able to visualize the parasites after the different stages, like if you can visualize after 35 hours or 72 hours or after removal of the drug, you can be able to visualize the microscope visualize the parasite with the help of the upright microscopes. So, in the step 1 you have to prepare the ring stage parasite. So, this is been the first step where the parasite culture, the mixture of different stages of the parasite is synchronized to the ring parasite containing RBCs. It has the following steps. So, the first step is that you have to prepare the ring infected RBCs, so that you can be able to then incubate those ring infected RBCs with the test molecules and say whether the ring is getting matured to give you the Chidon after 35 hours or not. So, for that you have to prepare the ring stage, and that you have to do with the help of the synchronizations. So, what you do is you take that 4 ml of a culture, which is where you have the peristemia of 5%. So, what is mean by the 5% peristemia is that the 5 RBCs are infected out of the 100 RBC which you are going to count, which means that is the 5% peristemia. So, you centrifuge the parasite culture at 72 G to pellet down the RBCs, and then you resuspend the RBC pellet with 4 ml of 5% sorbitol in distilled water and incubate for 10 minutes at room temperature and mix and shake it for 2-3 times. So, when you incubate this parasite culture with a 5% sorbitol, what will happen is that it is actually going to induce the lysis of the trophozoites as well as the Chidon containing parasites. So, it is actually going to lyse all the RBCs which actually contains the trophozoite or the Chidons, but it is actually not going to affect the RBCs which contains the rings parasites. So, then once you shake it for 2-3 minutes, it is actually going to perform its actions, and then you centrifuge the culture at 72 G, wash it for 3 times with media and bring the parasite to the 5% hematocrit. So, you know that the 5% hematocrit means that the 5% of the RBCs out of the 100% volume, which means the only the pack volume of the RBC is going to be 5%. The normal blood has a hematocrit of 50%, which means the 50% is the RBC component and the 50% is the plasma. So, in this case we are taking only the 5% hematocrit. You repeat this synchronization steps for 2-3 times, because you first time when you do the major chunk of the shy joint and the trophozoites are going to be lysed, and then you may still have some shy joint and trophozoite left. So, then your rings are going to be formed, and you may have also the rings of the different ages as well, because you can have the ring which is actually going to be formed after 2 hours, you can have the ring which is actually been of 8 hours. So, what will happen is within another 4-5 hours or 10 hours, you will see again, you will start seeing the trophozoites. So, to make it very precisely the synchronized culture, what you can do is you can do the synchronization with the help of the d orbital multiple rounds. Like first time you have done, then you again culture it for another 48 hours, then again you do it. So, if you repeat 2-3 times, you are actually going to bring only the ring containing parasites, and that is good enough for performing this assay. Then you calculate the parastemia after the Giemsa stain. The calculation of the parastemia we are going to discuss. So, parastemia you do not have to worry. Parastemia means the number of RBCs present in the 100 ml RBCs, means number of parasite containing RBCs out of the 100 RBCs, that is called as the parastemia, and we are going to discuss about how to calculate the parastemia in a subsequent slides. So, now once you have prepared the ring containing RBCs, then you have to do a preparation of the compound solutions. So, the test compound can be dissolved in the organic solvent at a concentration of 5 mg per ml. It is recommended to use DMSO as a solvent, as it has no significant effect on to the parasite growth. So, if it is a phytochemicals or if it is a water soluble compound, then you do not need to dissolve it into a organic solvent. You can simply dissolve it into a the aqueous media, and then you can just pass through with the 0.22 micron filters, so that it should be filter sterile. Otherwise, what will happen is as soon as you will add the compound, the bacteria will start growing. So, that actually is going to interfere with your assay system. And so, in the step 2 you are going to prepare the compounds, and then in the step 3 you are going to set up the assay. So, the parasite culture synchronized at ring stage by the d orbital treatment brought to the 1% parastemia. So, you remember that it was a 5% parastemia. So, whatever the parastemia you have, you bring it to the 1% parastemia with the 3% hematocrit, and in a total volume of 100 microliter, you take the 50 microliter of parasite culture, and it is mixed with the various concentration of the test compound like 0, 1.5, 3, 6.25. So, it is a serial dilution of up to 50 microgram per ml of a compound in 25 microliter, and in the remaining you can add the complete media. So, in a recipe, what you have is you have a 50 microliter of your the ring containing cells, then you actually add the 25 microliter of the compound, which is actually going to be of different concentrations, and then you add the 25 microliter of the complete media, and that is actually you going to incubate into the incubators and let them to grow for another 48 hours in the presence of the compounds. Since you are doing all these experiments, you have to add the chloroquine, which is actually a anti-malarial compound, as a positive control, and you can add the solvent as a negative control. So, negative control is like whatever the solvent you are using, if it is a aqueous solvents like PBS or buffer or whatever, then you can add the buffers. If it is a DMSO, then you have to add the DMSO, and that will be a negative control, and then you can add the chloroquine also, so that it will tell you that whether the assay was working or not, because the chloroquine is eventually going to kill the parasites. So, it is actually going to work as a positive control. Incubate the compound for 48 hours, then you monitor the appearances of the hemolysis or any other such effect. If there will be an hemolysis, because what happen is sometime you whatever the test compound you add, it actually does not have any parasitic effect, or it does not have a parasital effect, but instead of that it actually lyse the cells, and that is how actually it is going to interfere with your assay system. So, if that happens, then you, because if the RBCs are being lysed, then there is no way that the ring are going to be propagate to give you Shy joint, and then Shy joint is going to give you the merozoite, and then it is actually going to do a reinvasions. So, if you have any hemolysis, if you there will be any lysis of the RBCs, then you have to stop the reactions, and then you have to investigate which component of your assay mixture is actually giving you the hemolysis. If you can manage, because sometimes what happen is the osmolarity of the different compound is also interfering with the osmolarity of the complete reactions, and that is how it is actually causing the hemolysis. So, sometimes you may have to change your buffer systems and all that to take care of that. So, that is how you have to actually use the stop the assay, but otherwise if you do not see any hemolysis, then you can go on to the next step and you can monitor the growth of the parasite. So, in the step 4 you are going to monitor the step of the growth of the parasites. So, after the 48 hours you are going to make a smear, and the parastemia has to be determined after the GSB staining. So, using a oil immersion objectives. So, and these are the results what you are going to see. So, what you see is these are the healthy parasites. So, if there will be no inhibition of the compound, then what you are going to see is that the ring is actually getting converted into the all other stages of the parasites, like you will see the trophozoite and Shy joints and all that kind of thing. But if the ring is not being able to convert into the Shy joints, or even if it get converted into Shy joint, but the Shy joint is being dead, then what you will see is actually a RBC which is actually going to have the fragmented cells, like you are not going to see a clear nuclear membrane, and you will not be able to see a clear cytosol. Because for example, in this case you see that it has a very discrete cell, and it has two nucleus, and it is very clean and clear, but in this case all these cells are actually fragmented. So, these are actually the appearances what you will see when the parasite is dead actually, because that is how it is actually does not have the nuclear membrane, and the cytosol is also not very clear. So, that is how it will say that whether the assay is working or not. But if you want to calculate the growth of the parasite, then what you have to do is you have to count these cells under the microscope. So, you have to observe these cells under the 100x of magnifications, and then you have to keep counting how many number of RBCs are ring containing, how many RBCs are of trophozoite, and how many of Shy joints, and then you have to plot that, and using that particular data you will be able to determine the parastemia as well as whether the compounds are inhibiting or not. So, in the step 5, using that you can be able to calculate the IC50, which means the inhibitory concentration 50 of that compound. So, the number of Shy joint containing RBCs were counted against each concentrations. The Shy joint inhibition data from that in vitro Shy joint inhibition assay of the above compounds were fed into a specially pre-programmed Excel sheets. So, you can actually be able to calculate using this Excel sheet which is been available from this particular site. So, if you put all that Shy joint containing cells and put it into this particular Excel sheet, automatically it is going to plot the curve, and it will going to tell you the IC50 as well as the IC90 and all other kinds of parameters. You can add the chloroquine as a positive control, and that actually is going to give you the confidence that the assay was working, and there is no the flaws, or there is no issue with the setup of the assay itself. To determine the nature of the action, for example, the peristatic or the pericidal. So, peristatic or pericidal means that you have a ring containing parasite. So, what you will do is you will add the compound. So, if you add the compound, what will happen is that it is actually going to seize the the growth of that particular parasite. So, whether it is seizing because the parasite is not getting the enough nutrition, and in your the compound is somewhat interfering with the biochemical pathways, and that is how it is decided that okay, let us reduce the metabolic activities and remain as a dormant stage. So, in that case the parasite is going to be remain live, but it is not going to grow actually, because for growth it requires all those metabolic pathways to be in an activated state. The other condition is that it is actually converting the parasite into a ring containing parasite, but these rings are actually the dead. So, if they are dead, then they are not going to give you the Schizoin whatever you do actually, okay. But if they are live and if you remove the compound, which is, then they are actually going to give you the Schizoin, which means they can be able to complete their life cycle. So, if they are being able to the life and they will be able to complete, then the compound is going to be called as the peristostatic, which means it is actually only inhibiting the growth of the parasite. But if it is converting the parasite to a dead parasite, then the dead parasite will not going to complete its life cycle, and that is how it is actually going to be called as the peristocidal, which means it is going to kill the parasite. So, to determine that, what you have to do is in a 100 microliter volume, 3% hematocrit with 1% peristemia were exposed to the trial compound for 48 hours. After 48 hours, the parasites were washed twice with complete media, so that you can be able to remove the compounds, and then you incubate for another 48 hours in a drug free media. So, once you keep it in a drug free media, they are actually going to be free to grow. If they are live, they will grow. If they are dead, they are not going to grow. So, then you are going to prepare a smear, and the peristemia can be determined with the microscopically as we discussed, like you prepare a smear, then you count the the 1000 cells, which means the 10 fields actually, and if you count the 1000 cells, it will actually going to give you the statistically significant how many number of infected RBCs were present, and that is how way you are actually going to determine the peristemia. So, this is all about the one problem where we have used the light microscopy, and we have actually determined the anti-malarial activity of a test compounds. You can be able to modulate these kinds of assays, and you can be able to even use it for some other applications. For example, even if you see what we have discussed about the malaria parasite, but if you want to change the conditions and you want to utilize the microscopes, you can be able to change it in accordingly, and that is how you can be able to utilize it even for screening the compound for other assays as well. For example, you can use the even slightly derived version, and you can be used to microscopy to measure whether for even for cancer cells and all other kind of cells, whether the cells are growing or not. Now, we will discuss about the research problem 2. So, in the research problem 2, the scientists are routinely propagating mammalian cells, and now what the scientist has done, it has developed a new medium, which means he has developed a new media for cell proliferations. Now, what he wants to design an experiment to count the number of viable cells and number of dead cells, which means he has developed a new media, and now what he wants is, since he wants to test whether this media is good enough as it was already been established media compared to the established media, and whether the number of live or the dead cells are more or less in the case of when they are using this particular new media for propagations. So, experimental design is very simple. You are going to use a dye which is called as the trypan blue. So, trypan blue is a charged dye, and the viable cell exclude this dye to the presence of the membrane potential, whereas dead cell are actually going to accumulate the dye in the cytosol. So, what happen is you take the cells or you take the culture actually, and then if you add the trypan blue, what will happen is the trypan blue is actually going to be excluded by the cell, because trypan blue is a charged dye. So, it requires some receptor or some other active processes through which the trypan blue can be taken up by the cell. But if the cell is dead, because the cell is maintaining a potential across the membrane, and the dye has to neutralize that potential, then only the dye can be able to enter. But if the cell is dead, the trypan blue is actually going to be entered into the cell, and it is actually going to make the cell as the blue color. So, that is what you have to do. If you take the whole cell populations and if you stain it with the trypan blue, what you will see is that the some cells which have not taken up the dye and the some cells which are actually appeared blue. So, these are actually the live cells, because they are actually opposing the entry of the dye into the cell, because they have a active membrane potential, and that membrane potential is opposing the entry of the compound. Compared to this is a dead cell, and that dead cell does not have the required electrode the membrane potential, and that is how it is actually going to allow the entry of the dye, and the dye is actually going to accumulate into the cell. This is the hemocytometer, which actually you can use to count these cells with the help of the microscopy. So, the material what you require, is a glass slide, you require a cover slip, you require an inverted microscope with a phase contrast provision, like you require a phase plate and and then you require a hemocytometer. These are the multiple steps. So, in the step 1, what you have to do is you have to remove the cells from the cell culture plate, either by the trypsinization or by the 0.5% EDTA. So, this is the step 1, and then you plate the small amount of cells on a glass slide and cover them with a cover slip. Then what you do is you take the mix 50 microliter of cell suspension with the 50 microliter of trypan blue solutions. So, 0.4% trypan blue solution is already available from the different vendors that you can buy, and then you fill the hemocytometer chambers, observe the cell under the 20x objective using the inverted microscope with the face plate. So, viable cells appears colorless, whereas the dead cell appear blue or the dark colored. The hemocytometer is placed on the microscope stage and the cell suspension is counted. There is a V color notch. So, in the hemocytometer, what you have is, in the center you have a V color notch. So, on this V color notch you can actually through with that V color notch you can be able to you can be able to load the hemocytometer with the cells, and the cells are counted in a chamber, and that gives the number of cells. In addition, the blue colored cell can be counted to know that the number of dead cells. So, in this particular procedure, what you have to do is, the first you have to trypsinize the cells. So, when you add the trypsin enzyme, or you can use the EDTA, it is actually either going to chew up all the receptors what the cells are using to stick to the dishes, or it is actually going to destroy the calcium. So, whatever the mechanism, the cells are going to come off from the cell, from the dish, and then what you can do is you can just mix the 50 microliter of the cell with the 50 microliter of trypan blue, and then you load that onto the hemocytometer, and then you put it under the inverted microscope, and then you can actually visualize these cells within the chamber, and you can be able to count the number of blue cells and the number of colorless cells, and that is how you can be able to count the number of dead cells and number of viable cells. So, this is all about the theoretical understanding of this process. Let me take you to my lab, and we are going to show you all these procedures. And because the hemocytometer is a very if you very clearly see the inside structure, it actually has a different chambers, and then you have to do a counting in these different chambers, so that you will be able to count the number of cells, and then you ultimately you can be able to even calculate the concentration of the cell in per microliter or number of cells per ml as well, because that information is required if you want to plate a specific number of cells for a experiment. For example, if you remember when we were doing the when we were discussing about the phagocytosis experiments, the last time we said that you have to plate the 10,000 cells. So, if you want to plate the 10,000 cells, the first thing what you have to do is you have to put the cells into the hemocytometer, you have to count, and then you have to convert that value into the concentration, like 10 to power 6 per ml or 10 to power 8 per ml or so on, and then accordingly you have to dilute and calculate that how much microliter of the cell suspension I should take, so that it will give me the 10,000 cells per well. So that is also true for and all other assays like MTT assay and all other kind of assay what you do in your lab, where you have to do a counting. So let's understand how you can do the cell counting and how you can be able to determine what are the number of viable cells you have and what are the dead cells you have.
Hello everyone, in this video we will show how to subculture the cells and count the cells. First we have to remove the remaining media, then trypsinate the cells, then we will count the cells and see it. Now I will show how to do trypsination. Now I am going to add the trypsin to detach the cells. After cells are detached, we have taken into clean falcon. Then we have to centrifuge the cells. As these cells are very delicate, we have to centrifuge at 1500 rpm for 2 minutes. Now we have to remove the supernatant and resuspend the cells in fresh media. After resuspension, we have to count the cells. So I am going to take 20 microliter of this cell suspension and mix with the 20 microliter of trypan blue and count under new bar chamber. Before counting, we have to see how a counting chamber or hemocytometer look like. This is a typical hemocytometer, also called as Newbar chamber, which contains these squares in upper side and lower side, with each square having depth of 0.1 mm and area of 0.0025 millimeter square. Now I am going to put a cover slip on this chamber. Then I will add slowly cell suspension. Through capillary action, it will spread all over the squares. So we checked how many cells are there in all the squares. Now we have to count. How to count the cells? So here a typical Newbar chamber which contains squares, 5 squares. So we have to count cells in these squares. So each square is an area of 0.0025 millimeter square and total small squares 16. So total area of this whole square is 0.04 millimeter square. So the depth of this each well is 0.1 millimeter. So what is the volume? 0.04 into 0.1. So that is total 0.004 millimeter cube or 0.004 microliter. So say we have combined cells in each well. Say this is A, B, C, D. Here we have 100. Here we have 150. Here we have 110. Here we have 100 again. So the total cells we have to take average. That means 100 plus 150 plus 110 plus 100 divided by 4, total 4 squares we are counting. The average is 115. So 115 cells in 0.004 microliter volume. So how many cells per 1 ml? So that we can calculate simply 0.004 into 1000. That will give the volume cells per ml.
So, in this experiment the student have discussed different steps related to the cell counting and how you can be able to utilize the microscopes to determine the viable as well as the dead cells. They also have discussed how to calculate the concentration of your cells so that you can be able to use that information to you can be able to use that information to put the cells as per your requirements. Now, let us move on to the next problem. So, the next research problem is that Mycobacterium tuberculosis H37RV is inhibiting the interaction between the phagosome maturation, and now you have to design a suitable experiment to study the interaction of the phagosome with the lysosomes. If you remember, when we were discussing about the phagocytosis in our previous lecture, we said that the phagosome is formed when a particle is being taken up by the cell through phagocytosis. So, once a cell is being taken up a particle, like for example a bacteria, in this case the Mycobacterium tuberculosis, this particular particle is going to be phagocytosed with the help of the pseudopodia, and then ultimately it is going to be internalized. So, in the internalized thing what you have is this is the bacteria what you have, and it is being internalized by a membranous structure, and this structure is actually being called as the phagosome. And then eventually what happen is that you have the lysosomes in the cell. So, all the immune cells have the lysosomes. So, then these lysosomes actually fuses with these phagosome, and this is a very complicated and complex process through which the phagosome actually interacts with the lysosomes and eventually developed, and the lysosome fused, and then when the lysosome is fused it actually causes the release of its content, and that is how the lysosome is actually going to release its content, and that is how lysosome is going to digest this material. So, lysosome is lysosome's inner pH is around 2, and it also contains the different types of enzymes like hydrolases and proteases and all those kind of enzymes. So, all these enzymes are actually processing the cell, the bacteria, whatever the immune cells have the engulfed, and that is how it actually degrades the bacteria. But what happened in the case of Mycobacterium tuberculosis, it actually interfered in this whole process and eventually survived inside the macrophages, and that is how it actually propagate within the immune cells, and that is how they will be able to rescued or survived from the action of the immune cells. So, what we supposed to do is we have to supposed to design an experiment to study the interaction of the phagosome with the lysosomes. So, in this experiment what you have to do is you have to first form the lysosomes. First you have to form the phagosomes, and then you have to form the lysosomes, and then you have to mix them together, and then you have to ask them whether they are actually fusing or not in the presence of tuberculosis or bacteria or not. Or suppose you give the phagocytosis, the Mycobacterium tuberculosis bacteria itself for phagocytosis, and then you study these interactions. So, we have already discussed about the experimental design where you have to first form the phagosomes, then you have to also form the lysosomes. And how you are going to do a phagosome? You are going to form the phagosome. What you can do is you can simply allow the cell to do a phagocytosis, and phagocytosis if you do, it is actually going to form the phagosomes. How you can be able to generate the lysosomes? What you can do is you can ask a cell to drink some liquid. For example, you can just go with the process of pinocytosis, and if you do so, whatever liquid you will give, that will enter into the lysosome. So, for example, if I take a fluorescent dye and if I add that dye to the cell culture media, eventually what will happen is the lysosomes, whatever is going to be formed, are actually going to be fluorescently labeled, because this liquid what we are taking is actually going to be filled into the lysosomes. Once these two are ready, then you can mix them and ask whether they are fusing with each other or not, because if the mycobacterium has any effect or it is actually having some factors which are interfering into this, it is actually going to it will not allow the phagosome to interact with the lysosomes. So, the material what you require the methanol and acetone, that is you require for fixation, you require the PBS, that is only for washing purpose, then you require the Triton X100, that is for permealizations, you require the BSA, you require the epifluorescence microscopes, you require the 1 micron latex beads, you require the Philippines, and then you require the rhodamine dextran. So, rhodamine dextran is a fluorescent dye which actually going to allow you to form the lysosomes. So, the procedure what we require, the procedure is that in the step 1 you are going to do the identification of the phagosome, or you are going to prepare the phagosomes which you can as per the provided by your own material. So, what you can do is you can take the J774, these are the macrophages cells. So, these are the immune cells which you can use for phagocytosis. So, these are the macrophages. Cells are cultured in a DMM media containing 10% FBS and 1% cocktails. Then you remove the cells from the cell plate by trypsinization or EDTA. You plate the 10,000 cells on cover glasses and incubate in a 24-well dish. Then you incubate the cells overnight and allow the cells to attach to the cover glasses. Then you wash the cells with DMEM. Then you prepare a suspension of the latex beads like 10 to power 6 latex beads per ml in a DMM media. So, what you have to do is you have to maintain a ratio of 1:10 in terms of the number of cells what you have taken and the number of beads what you have taken. So, you are going to provide the 10 beads for every cell to feed, and then you remove the media and add the suspension to the centrifuge. And so, basically what you are doing is in the step 1 you are basically doing the phagocytosis, and that is how it is actually going to eventually form the phagosomes. So, these are the wash the cells with this and that, and then you fix the sample with the acetone and then you stain it with the Philippines. So, that actually is going to allow you to identify the phagosomes, because wherever you will going to see the object being encircled by a blue color fluorescence, that is actually the phagosomes. And then you mount these cells into and you can be able to identify the phagosomes. Then step 2, you are going to do a labeling of the lysosomes. So, what you do is you plate the cells on a cover glasses in a 24-well plate, grow them with a 100 microgram rhodamine dextran overnight in a DMEM plus 10% FBS plus antibiotic cocktail. So, what will happen is when you are going to grow the cells in a media which contains the rhodamine dextran. So, rhodamine is a fluorescent dye and it is coupled with a dextran. So, dextran is nothing but polysaccharides. So, what will happen is the cell will start going to eat these rhodamine dextran. So, when it will eat, eventually all the material will end up into the lysosome, because you know that the primary function of a lysosome is to digest the material what you have ingested, or whatever the the material is being ingested by the cells, because ultimately you have to you have to digest all that material, generate the constituents material, like suppose you digest the protein, then it is going to generate the amino acid, and then these amino acids are going to be supplied to the cell for its propagations or for nutrition. So, whatever the media you are using, you use the same media, you add the rhodamine dextran and then you let them to grow for overnight, and that is how the rhodamine is going to be end up into the lysosomes. you wash the cells with PBS and then you are going to chase the sample for 1 hour. So, what is mean by chasing is that you remove the rhodamine dextran and then you let the things to grow without without rhodamine dextran. So, what happen is if you do so, the last drop of the rhodamine dextran will also going to be end up into the lysosomes, because eventually everything end up into the lysosome, because that is the way the cell has devised the mechanism. So, that whatever it drinks or whatever it eats, it ends up into the lysosome, so that it will get digested and you can be able to use the nutrient coming out of these digestions. Then in the step 3 you are going to set up the fusion assays. So, you add the 10 microgram 1 ml latex in 0.5 ml media and spit at 1000 G for 2 minutes. Then you incubate for another 5 minutes in water bath. So, when you do that in incubation in 5 minutes for water bath, it is actually going to induce the phagocytosis of these beads. And then you remove the beads and wash them twice with the PBS containing 37 degrees Celsius. The media is removed and fixed with the paraformaldehyde. Then your slides were visualized in the microscopes. So, in the incubation assays, what you are going to do is you already prepared the lysosome in the cell. Then what you do is you add the latex beads to these cells and allow them to phagocytose. And once they are phagocytosed, the phagosomes are being formed. Lysosomes are already been there because they are already been fluorescently labeled. And now what you can do is you can just do these incubations for the multiple time points, like you can do 5 minutes, 10 minutes and up to 1 hour. In this process, the phagosome will meet all the lysosomes and the lysosomes are going to be fused with phagosomes. So, what will happen? When you visualize these cells under the microscopes, what will happen is that if you observe the cells in a bright field and look for the beads onto the cells. So, what you are going to see is you just visualize the cells under the microscope, then you look for the beads under the bright fields. To observe the cells in the fluorescent microscope with a UV filter, if the beads has a blue fluorescence, like in this case, this is the bead, right, and it has a blue fluorescence around it, which means this is actually a phagosome which is being formed already. Now, once you saw the phagosome, then what you can do is then the cells can be visualized through a red channel. So, what you are going to do is first you select the bead what you want to visualize under the bright fields, then you go with the blue channel. So, in the blue channel it is actually going to give you a blue fluorescence around it, and then you go into the red channel. So, once you go into the red channel, what you will see is that this blue colored signal is also having a red color signal, which means if you have the double signal, like if you have the blue color ring and then if you have a red color ring around it, which means these are the phagosomes which are actually having the lysosomes as well, which means here the phagosome is being interacting with the lysosomes, and that is how the phagosome has received the content from the lysosomes. So, that is how you can be able to conclude that the phagosome is being now matured and it has formed the phagolysosomes. How you are going to analyze this? A typical phagocytosis of bead will represent by the appearance of the bead in the phase data, which means this one, and the same bead will be circled by a blue fluorescence from the Philippines. If the bead has also blue fluorescence ring and it further encircled by a red color ring, indicate that the interaction of the lysosomes and phagosome is happening. You might see that some of the beads, like for example, this bead is also being formed and this bead does not have any phagosomes. For example, in this case you see this is the bead which is actually being internalized and you have a blue color fluorescence, but for this particular bead you do not have any signal into the red fluorescence, which means this bead is only forming the phagosomes, but these phagosomes are not interacting with the lysosomes. So, that is how you can be able to study the interaction of the phagosome with the lysosomes, and what you have done is you have simply allowed the cells loaded with the lysosomal markers or the fluid markers, so that the lysosomes are being labeled with a red color fluorescent, and then you allow the cell to phagocytose the material, and once it will phagocytose, it will going to form the phagosomes, and then it is actually going to interact with the lysosomes, and wherever the phagosome is going to interact with the lysosomes, you are going to see a bead which is circled by a blue color ring, and blue color ring is further encircled by a red color ring. So, if you have two rings, red and blue, so that is the place where the phagosome is being interacted with the lysosomes. So, with this we would like to conclude our lecture here, and in this lecture we have discussed the different experiments what you can be able to perform with the help of the microscopy, and I hope you might be able to design few more experiments after looking at the potential of these particular techniques, and it may help you in designing the new experiment for your own work. So, with this I would like to conclude my lecture here. Thank you.